Hey everybody, so today I'm going to have you do your first Bell quiz, and what you'll need to do is get your notes packet out and turn to the very front page. Okay, and on the very front page of your notes packet you have a bunch of squares. Okay, let me show you what I'm referring to. So here is a notes packet from a previous student. It already has some Bell quizzes in there, but I'm going to have you pick the first box in that top left-hand corner and put today's date, right, which is March 11th, and then you're going to answer these two questions in that box. Okay, you don't need to rephrase the question, just simply write number 1, what's the answer, number 2, what's the answer. Now, in order to get credit for your Bell quizzes, you just need to answer them, and you got to answer them based on the question. So if you're absent, I do allow you to work with your partner, but I don't want you to use your notes. Okay, I want to go off of your memory. So if you were gone, your partner's going to have to help you out as far as teaching what this material is. Okay, so 2 questions you're going to put in that top left-hand box. All right, what are the 3 parts that make up a nucleotide? And number 2, what are the 2 types of bonds that are present in a DNA double helix? All right, there's 2 of them. And where can you find them? So I need you to tell me what the name of it is and where would you find it. All right. So we'll go ahead and pause this video and let you do your Bell quiz, and then we will go over those answers. Okay, so here are the answers to your Bell quiz. First off, the 3 parts that make up a nucleotide. You got your phosphate, you got your sugar, your deoxyribose, and it has that pentose shape, and then you have a nitrogenous base. Okay? And you might have put A, T, C, or G there, which is awesome, because there's 4 different types of nitrogenous bases, and you could have picked any of them. Or you could just wrote nitrogenous base. Okay, so that's number 1. Number 2 is what are the 2 types of bonds and where do you find them? All right, so one of the bonds, and it doesn't have to be in this order, is hydrogen bonds. All right, hydrogen bonds are in the middle, dead center, okay, between the rungs of the ladder. So they are between your nitrogenous bases. That's why you got those dotted lines, kind of symbolize there's a different bond there. Something to remember those hydrogen bonds, they are weak, and we like to rip them apart when we are replicating, making a new DNA strand. So there's your hydrogen bonds. And then over here, between the phosphates and sugars and the nucleotide, phosphate, sugar, nucleotide, so between these molecules, you have covalent bonds, and those are your really, really strong bonds. So covalent bonds hold a nucleotide together. Okay, hydrogen bonds hold nitrogenous bases together. Okay. All right, now, in order to score this, as long as you answered the questions, you automatically get 5 points. So give yourself a 5 out of 5. And if you happen to get both questions right, you and your partner, then you get a bonus point. You can give yourself a 6 out of 5. Okay, next, I need each person to grab a Chromebook. Because with this Chromebook, we're going to take our first quiz in biology. It's going to be over the DNA structure. So here we are at the home page, and there's 2 ways you can get to it. So one way is if we go to the daily schedule, and if I scroll on down here to Friday, March 11th, you can see here it says click here to take your quiz. Okay. So I click here, and then it's going to pull up your DNA structure quiz. And then obviously another way you can get to it, let me extend this over, is if you click on quizzes. And once you click on quizzes, you can see here as well. So I'd like you to take this quiz. You'll have about 5 minutes. So grab that Chromebook, get logged in, and take this quiz. Good luck. Okay, now before we start into DNA replication, which is the notes on page 5 in your notes packet, I just want to watch a quick little video, just reviewing DNA structure. You just took your quiz on it. And just go over some key parts, and I want to pause it and just do a couple things, okay? So we're going to play that now.
This is a 3D model of a DNA molecule. It is made up of 2 strands that form a double helix.
So hopefully you're already looking at that and going, yeah, that is 2 strands of a DNA double helix all twisted together. And this part, okay, kind of the darker pink, that's the backbone. That's your sugars and your phosphates. And then the middle part, it's yellow and light pink, that's your nitrogenous bases, right? Because DNA is just made up of a bunch of nucleotides.
If we unwind the helix, we can see that each strand is a chain of building blocks called.
All right, so looking at this, it reminds us that DNA is just a chain of nucleotides. So here we are again. It's just cool to see the molecular structure of it, and you know what this is, right? There's that sugar-phosphate backbone. Sugar-phosphate backbone, here's the nitrogenous bases. And if this was an A, then you got to know this has got to be the T. This is a C, you got to know this has got to be a G, right? And we'll keep going.
Let's pull out these 5 pairs of nucleotides to take a closer look at their parts. These diagrams show the chemical structures of the nucleotides. The nucleotides are linked together. They are linked one way on one strand and in the opposite way on the other strand. Because the strands are arranged in opposite but parallel ways, they are called antiparallel. All nucleotides in DNA are made of the same basic parts. They include deoxyribose sugar molecules, which form the backbone of each DNA strand, and nitrogenous bases, which have 4 different types in DNA. Nucleotides are linked by chemical bonds called phosphodiester bonds between their sugar molecules and phosphate groups. The phosphate groups.
Now those phosphodiester bonds, okay, we are referencing these, and those are your types of covalent bonds. Those are the ones that I need you to remember. All right, so we know we've got the covalent bonds that are here, right?
Are shown as the circles highlighted on this diagram. Here is the chemical structure of a phosphate group, which has a negative charge. This is the chemical structure of a deoxyribose sugar. The carbon atoms at the 5' and 3' positions of the deoxyribose sugar form bonds with the phosphate groups. The 5' and 3' positions are used to show the orientation or direction of a DNA strand. During DNA replication, enzymes build DNA strands by adding nucleotides in the 5' to 3' direction. DNA has 2 kinds of nitrogenous bases.
Okay, so we're going to pause it there for a second. So it just said that it's going to add DNA nucleotides according to the 5' to 3' direction. So let's look here. So this is a 5', this is a 3'. So when this DNA, when those hydrogen bonds get broken apart, and it creates 2 separate strands, there's a cool enzyme that's going to come in. It's called the Dna Polymerase, and this enzyme is going to come in and it's going to add nucleotides. Okay, according to the rules, like A, it'll add the nucleotide that has a T, and C, it'll add the nucleotide that has a G. But it's going to go in a certain direction, right? So when you look here, you have the 5' here to 3', and then on this side you have the 5 to the 3, which means the enzyme, when it comes in, this enzyme's going to work this direction, and this enzyme's going to work this direction. So the enzymes are actually working opposite directions. Okay? So that's kind of why it's important of what the 5' to 3' is. Now this picture that you guys have colored over on page 4, let's just go ahead and add where would the 5 and the 3' go. Okay. So the 5', it's always going to be at the phosphate end. So you can put a little 5 here, all right, a little like tick mark. So you got the 5, all right, this is your 5' side. And then if we follow this, one end is the 5', and if we followed it, then this end would be the 3'. Okay. All right, now there's your sugar. All right, the sugar end, it is going to be the 3' side. So you can put a 3 here. And we're going to follow this one down until here you have the 5'. Okay, so you always, on a DNA strand, you always have a 5' end and a 3' end. And then the complementary strand is the opposite, which is why they call them anti-parallel, because they're the opposite, but yet they still match up according to the rules, A with T and C with G. Yeah. All right, we're going to go back to that video now.
The first kind are the purines, adenine and guanine. The second kind are the pyrimidines, thymine and cytosine.
All right, so those are just some extra terms, purines and pyrimidines. You don't necessarily need to worry about that. But what you do, could be helpful to notice is that T's and C's have similar shapes. Just like your A's and G's are a double ring, so they have a similar shape, right? So you should never see a single ring paired up with another single ring. That's a problem. Just like you should never see a double ring paired up with another double ring. All right, so it's kind of an easy way visually to get an idea if they are paired up correctly, right? So you should have a single ring always paired up with a double ring.
The bases on one strand of DNA bind to bases on the other strand through chemical bonds called hydrogen bonds. The bases bind in specific ways. Cytosine binds with guanine, and adenine binds with thymine. Bases.
So I like that you can see A and T requires 2, okay, hydrogen bonds, and once again C and G require 3 hydrogen bonds.
That bind to each other are called complementary, meaning matching. If you know the sequence of nucleotides on one strand, you can figure out the sequence of the other strand by knowing which bases are complementary. For example, this top strand has the sequence A, C, G, T, T when read in the 5' to 3' direction. And the bottom strand has the sequence A, C, G, T when read in the 5' to 3' direction. The bottom sequence is made of bases complementary to those in the top sequence, but in the reverse order. The 5 pairs of nucleotides we looked at are only a small part of this DNA molecule, but the components and relationships are true for all DNA.
All right, so what I'd like to do is we're going to go to your notes packet, and what I'd like to see have happen is let's practice doing those base pairing rules. Okay, putting A's, T's, C's, and G's together in the complementary way. So I need you guys, go to page 5. All right, so here's the DNA replication where we're at right now. And let's just go to the very bottom right here. Okay, so it says put the correct base with its complement. When A goes with T and C goes with G, we call these the base pairing rules, because it's when the bases pair up and there's certain unique rules. So here is the parent strand. All right, that's the original strand. And now your job is to figure out what would be the complementary strand, what pairs up to it. So I kind of got it started for you, okay? A is going to pair up with T, right? Those base pairing rules. G pairs up with C. All right, what I'd like you to do is finish giving me the rest of that complementary strand. So just pause this video and finish filling that part out. All right, so I went ahead and drew out that complementary strand. So check your work, see how you did with mine. So T and C was already done for you, but then the rest of the complementary strand you should have had A, G, T, G, C, G, T, A, G, T, G, A, C, C, G, C, T, A, T, G, T, A, C, and then T, G, G. Hopefully that was easy for you. But what I'd like you to do right now is I actually want you to label the parent strand and the complementary strand. Okay, so we're going to put a big P by this one because this is our parent strand. That's the original strand we have. And then this would be called the complementary strand, right? And we're going to learn what I'm going to teach right now is, once again, the enzyme that'll come in and it'll build it. It'll add these 2 nucleotides. Okay. All right, so let's jump back to the PowerPoint.